Today I'm going to talk to you about some of our antibody responses studies to SARS-CoV-2 that we've been doing in Philadelphia since the pandemic has begun. So my lab primarily focuses on influenza virus, at least up until 3 or 4 months ago, and we're very interested in defining where human antibodies latch onto this virus. We're very interested in identifying antibody types that are associated with protection versus susceptibility. And we've been studying variation among different humans. Different humans have different types of influenza antibody responses. And at the end of February, beginning of March, we very quickly shifted over to studying SARS-CoV-2. And this was not a huge jump for us because influenza viruses change, and we are very used to changing our antigenic assays each year as flu viruses change. And so at the end of February, beginning of March, we began to clone constructs to express this red protein here called the spike protein, which is probably one of the major targets of antibodies against this new virus. And so we had been making constructs to express these proteins when a collaborator of mine and colleague, Florian Kramer from Mount Sinai, published a preprint showing that he basically produced a very similar construct, and it was available. And so, you know, in a time of a pandemic, you leave your pride at the door. I sent this email on March 17th, 3:45 p.m. Hey, Florian, can you send us those plasmids? And this is the response I got 3 minutes later. Sure, they're in the mail. And so this is what science looks like during a pandemic. I wanted to start with this slide today for two reasons. Number one, I want to thank Florian Kramer and his group for designing these constructs and distributing them to my lab and others across the world. But number two, I want to highlight that this is how science could be. There's very few good things that are going to come out of this pandemic, but the way we actually do science and respond, and the way we've responded to this pandemic, it has been transformative. It's been a real honor to be part of this collaborative effort to try to respond to this virus. So the trainees out there that are watching this is the future. This is how science is going to be done for everything 10 years from now. So here's that spike protein. It showed you a variant that had a bunch of red proteins on the outside. This is what the protein looks like if you zoom in, and this is what we call the receptor binding domain. This little part here. So the spike protein, what the spike protein does is it basically attaches virions onto host cells, and it does it through interactions with this part of the spike. Now, this is a very attractive target for antibody assays because this part of the spike is very specific to SARS-CoV-2. It's very dissimilar to previously circulating coronaviruses. And so we've now gone on to develop a bunch of different antigens to test for these serological assays, but this still is our gold standard that we use, and this small bit of the spike protein called the receptor binding domain. So we are in the beginning of a pandemic. What do? Well, we turn to our ELISA systems, and we basically use these assays called ELISAs that we have been using for years now for flu. And instead of having flu antigens into those assays, we simply put in the SARS-CoV-2 spike receptor binding domain protein. I think this is the technical part of the symposium, so most of you are probably familiar with these assays. But we basically we coat plastic wells with the protein, and then what we flow on top are human antibodies, human samples, serum, and then we come in with the detection antibody, so an anti-human IgG, an anti-human IgM, IgA, and this detection antibody has something that will give a signal that a plate reader can read. And so here's an example of a validation assay of using ELISA plates coated with the receptor binding domain of SARS-CoV-2. In this case, we have tested 834 samples these are serum samples from 834 individuals before the pandemic. And here's 151 people, serum samples from 151 people who had PCR confirmed SARS-CoV-2 infection and went on to recover. These are actually, in this example, donors for plasma therapy trials here at Penn. And what you can see is that there's very little background, which is good, very few false positives. These guys are actually interesting. Some of us do walk around pre-pandemic with levels of cross-reactive antibodies, but it's pretty rare. And you can see most of us seroconvert. 90%, 95%. And in fact, if you wait longer and you look at some of these negative folks, they become positive if you take their samples at a later time point. This is something the Sinai group has shown. As I said, we have IgG assays as well as IgM, IgA. As many of you know, IgM comes up much earlier and then kind of contracts. And so there's much more variability with IgM, and that's simply reflective of how far along and how many days have gone by post-infection for these 151 individuals. So this assay, this receptor binding domain assay, looks pretty darn robust. And as I've said, we've gone on now and we've optimized assays with full-length spike. We've optimized assays with N protein, and we're working on several other assays right now as well. Now, I know during Paul's talk this morning, there's a lot of talk about neutralizing antibodies, non-neutralizing antibodies. This is an important discussion. But what I'll say is that there's a very good correlation between receptor binding domain antibody titers and neutralization. So we've been working with Paul Bates and Sarah Cherry. And so Paul Bates has this great assay. It's a VSV pseudotyped assay. So it's a VSV virus that basically has SARS-CoV-2 proteins on the outside, the spike protein on the outside. This has the advantage that you can work under BSL-2 conditions. And you can see here, as we plot receptor binding domain IgG titer versus BSL-2 pseudotype neutralization titers, there's a very nice correlation. And when we actually go into the BSL-3, and these are studies with Sarah Cherry, we test those same samples and do a bona fide neutralization assay. So we ask how well antibodies can actually block infection of cells with live virus in the BSL-3 lab. Again, you see a very nice correlation. Those samples that are negative in both assays, and positive as antibody titers go up, neutralization assays go up. So what do we do with these assays? So the first thing that we did, we established what we called the Penn healthcare worker SARS-CoV-2 serological study. And I want to go back in time and go over sort of our thought process as we set up this study. Now here are our case counts of Philadelphia, starting in mid-March and going on until June in this case. And this is when the healthcare project was conceived on 3/19. The good thing about emails, you can go back and you can kind of look backwards, and it's interesting sometimes to see how projects were, how they've evolved. And there's about 15 emails that went back and forth between myself and other collaborators on March 19th. And you could see we were very early in the pandemic in Philadelphia and really in our country at that point. And let me tell you, this has been a colossal effort, but we have set up a study, as I'll show you in the next few slides, where we've enrolled 2,000 healthcare workers. And within a month of conceiving this study, we were able to start testing. Now, keep in mind, in those early days, and I think we all remember those early days, we knew virtually nothing. And so what were we trying to address with this study? How many of our healthcare workers had already been infected? Does PPE work against this virus? We had no idea. Was everyone infected? We knew the virus was in our community undetected for a long time. Were all the healthcare workers infected, or did the PPE actually work? Are we going to run out of hospital beds, masks? I mean, these are the kind of questions that we were asking in these early days. We had no idea what this pandemic would look like moving forward. And so I want to just take a moment to thank the funders of this study. So the 76ers and Joel Embiid led this group, and then the Philadelphia Eagles came in as well and put some money into this effort. $1.7 million in total was donated to Penn Medicine for these studies. And let me tell you, not only was this a morale booster, but this allowed us to just pursue these studies without any hesitation. We cut no corners. And I like to show this picture of me and Joel. This was obviously pre-pandemic. In all seriousness, I do want to meet Joel one day to personally thank him, because this study really launched what was the backbone of all the serological studies that we're doing here at Penn. So what were our objectives? And I want to say that I'm a co-PI with Anne Frank, who has really led all of the clinical activities of this study. Our primary goal was to determine the rate of asymptomatic and symptomatic SARS-CoV-2 exposure of healthcare workers. How many of our healthcare workers are getting exposed? How many of them are symptomatic? How many are asymptomatic? Secondary goal, we wanted to determine if serology in and of itself can identify active asymptomatic infections. The idea was if we could perform serological assays in real time, that we might actually catch infections before an individual even knew they were infected. Another secondary goal was to collect serum and PBMCs to characterize the immune response in these healthcare workers, asymptomatic and symptomatic infections. So as I said, we enrolled 2,000 folks into this study, and this here in panel C shows the number of samples that we collected on each date, and in black are the negative samples, the samples that did not have antibodies against SARS-CoV-2, and red are the positive samples. So good news, we screened 2,000 people and only about 2% of these individuals were seropositive. That is a very low rate of seropositivity, which is a good thing because it indicates that the PPE that was used very early in the pandemic was doing its job. Now you can see here, this breaks down, we had 41 individuals out of the 2,000 that were seropositive as they entered our study. And out of those 41 individuals, you can see that there were different degrees of IgG and IgM responses, which is interesting from a basic immunology standpoint. Now, I told you that one goal here was to identify active and recent asymptomatic infections. And I don't think I said this from the start, but in order to be eligible for our study, participants could not have had diagnosed COVID-19 disease in the past. So really, we wanted people to come into the study with a blank slate. And so those 41 individuals that we identified seropositive at the beginning, they were essentially asymptomatic. They did not know that they were infected. And so out of those 41 seropositive individuals, we performed NP swabs and PCR analysis on all 41 samples, and 6 of those individuals had active or recent infections. They were PCR positive. And as we heard from the first talk this morning, it's unclear. It does seem that some people can continue to shed RNA without having virus. But this is sort of remarkable, right? 41 individuals, they're seropositive. 6 of them were walking around at least with an NP positive signal. And this was good in the sense that we could then have these folks quarantined so that they would not spread the virus within the hospital. Now, this also allows us to do some pretty interesting immunology, as I'll show you in the next slide. To summarize, we had 2,000 folks enrolled, 41 seropositives. Now, we continued to collect samples every 2 weeks, and we went on to identify 20 additional new cases over a course of 2 months of this study. Okay, so these are people that were seronegative when they enrolled in the study, and then they became seropositive over the course of the study, 20 of them. 10 of these additional participants that seroconverted, they were without symptoms altogether, just like the folks that were positive as they enrolled into the study. There were 10 additional participants that actually became symptomatically infected and went on to seek an NP swab outside of our study. And so you can say our overall exposure in the last 3 months of our cohort is probably about 3%. And we don't really know the history of these guys and these men and women that came into the study seropositive from the start. But what we can say is that over the course of the study, it's about half of the individuals that became seropositive were symptomatic and the other half were asymptomatic. And I think that's really interesting because we have a very poor understanding of ratio of symptomatics versus asymptomatics. We need to study this further, but I think this will prove to be very interesting. Now, as far as basic immunology, this is a gold mine. Not only are we collecting samples every 2 weeks, but we are even doing more extensive collections on those that are either ELISA positive or NP positive. So let me walk you through this. And so here on this graph, these are people that were seropositive and NP positive at that first visit. They had either active infection or very recent infection and had seroconverted at that initial visit. And what you can see over time, each one of these colors is an individual. The antibody response, for the most part, looks pretty darn stable. Now, here's the second group. These people were ELISA positive, seropositive, but NP negative during that first visit. These are individuals that probably got infected sometime in the previous 3 weeks when the virus was still rising in Philadelphia, but not our study had not yet began. And again, what do you see? You see pretty stable antibody titers. This is all good news, folks. Look on the third graph. So these are participants who seroconverted. This is super interesting for us because we actually have a timestamp of when these individuals were infected. They came into the study seronegative. They became NP positive, infected, and what you can see is here you can actually see that primary phase of the immune response where the titers are increasing. And then, as you might expect from normal immunology, you have titers that decrease a little bit after that primary phase. But what we suspect is that these individuals will sort of fall into this long, stable immune response that we see in these other two graphs. So this will really allow us to look at the longevity of the response. A lot of these questions when we think about vaccination and the first talk on vaccination, how long-lived are responses? Look, we've been following these people for 2 months, and it seems like these responses are pretty long-lived in most individuals. And we'll be able to look at asymptomatics versus symptomatics and if antibody longevity is different in these two groups using this cohort. To summarize, very low SARS-CoV-2 seropositive rates in healthcare workers in our Penn system. Half of the healthcare workers who were converted during the study were symptomatic. PPE works, okay? And that should be reassuring for these brave men and women who are literally putting their life on the line, working with COVID-19 patients. This cohort will be interesting to follow up, and we continue to follow these individuals to study the antibody responses. And as I mentioned, we have PBMCs as well, and we're going to start to interrogate T cells and B cells in these individuals as well. So I just, I framed what I just told you about the healthcare workers as low seroprevalence. Well, relative to what? So what is the overall SARS-CoV-2 seroprevalence in Philadelphia? And, this is a tough question to answer because it's difficult to get blood samples in an unbiased manner during a pandemic. You might have seen on the news that there's been large studies in New York where blood samples were taken at grocery stores. That's a neat idea, but, some people don't go to grocery stores during the pandemic. That might not be the entire population. There are several populations that continue to seek medical care during pandemics. And one of these groups are pregnant women when it's time to deliver a baby. There's not many pregnant women that decide to stay at home and deliver their baby via Zoom, right? Most women, when it's time to deliver a baby, go seek medical attention. And so we thought that this would be a unique population to monitor, to measure seroprevalence in Philadelphia over time. And so we've been testing leftover blood samples from pregnant women delivering in 2 of our hospitals in Philadelphia. And we started this April 4th, and these studies are ongoing, but the data I'll show you today is from April 4th to June 3rd. You can see we have 2 hospitals. This is HUP here. This is Pennsylvania Hospital, which is downtown. Those of you that are not familiar with Philadelphia, this is the city. And you can see that we had samples basically all across the city as well as surrounding areas in both Pennsylvania and New Jersey. So we found 6.2% of the women that we tested were SARS-CoV-2 seropositive. They had antibodies against this virus. They had been exposed to this virus. You can see in this case, we tested pre-pandemic samples from pregnant women collected before the pandemic. I think these samples were collected in 2015, 2016. And we have samples here from the women from April, May, and June of this year. 6.2%. IgG and IgM, most of these samples were IgG, some were IgG and IgM double positive. We were able to look at this seroprevalence over time. And so what you could see this is a sliding scale, a 7-day scale. You could see when we started the study, it was about 5%. And look what happened. We got to about 8%, and it seems to be pretty leveled off here at 8%. This is interesting. We plan on continuing to measure seroprevalence in this population over time. There's been a lot of talk of herd immunity and how we're going to get out of this pandemic. I want to take the same data here and put it on a different scale. Okay, this goes up to 9. This goes up to 100. Okay, so there has been a real raise in seroprevalence, and you really appreciate it when we have this zoomed up view. But the level required for herd immunity is probably, as Paul Offit said, something like 50%, and we're nowhere close to it, right? Now, do pregnant women represent immunity in the rest of the population? We don't know. We think they probably do. You can imagine it going either way. Pregnant women continue to seek medical care, not only when it's time to deliver, but also for routine prenatal visits, for example. Pregnant women typically have other young children in the house. These things would contribute to higher seroprevalence rates relative to the rest of the population. On the other hand, you can imagine pregnant women are more careful, and so it could go either way. But we think that pregnant women probably represent population level immunity pretty darn well, and we need to do these comparative studies. But if they do, and if seroprevalence rate in Philadelphia really is 8% right now, we are nowhere close to herd immunity. And we're not going to get to herd immunity through infection. We better hope we don't, because that will be a total disaster. In order to get from 10% to something like 50%, well, I'm not a mathematician, but I can even do that math. That's 5 times the number of disease and deaths that we've already seen in our city. So the real way to get to high levels of herd immunity are through vaccination, as we heard about this morning. Here's the finding that almost made me fall out of my chair. When we broke down the seropositivities by race and ethnicity, we saw major differences. And so one good thing about studying pregnant women is that pregnant women of all demographics continue to deliver babies. And what you can see here is that Black women have about a 10% seropositive rate. Hispanic and Latino women have about a 10% seropositive rate. White women and Asian women, a 1 to 2% seropositivity rate. That is remarkable. You got to remember, antibodies to SARS-CoV-2, serology, what does it measure? It's a stamp of exposure. Black women and Latino women have been exposed at tenfold higher rates compared to white women and Asian women in the city of Philadelphia and surrounding areas. That is remarkable. And what are the root causes? We don't know, and it's something that we are going to study. But we think that this probably has to do with the inability to socially distance. And this is probably tied up to density of living, things like socioeconomics, which is all a result of systemic racism that has occurred in our country for decades and decades. And so this is going to require quite a bit of further analysis, but this is remarkable. You know, it's been noted that COVID-19 has disproportionately affected African Americans and Latino communities in our country. It's been suggested that this is due to less access to healthcare, for example, and things like differences in comorbidity. All that might play a role, but look at this. Just pause and look at this. If there's a tenfold increase in exposure in some of these populations, well, it's no a doubt contributing to the increase in COVID-19 disease and deaths in these populations. This is something that we have to learn more about and fix. So I want to summarize this second part and leave some time for some questions. So we found this overall 6% seropositivity rate, with it rising as we should expect as cases have continued to increase, and we see it stabilize at about 8% now as cases have decreased into the city. And we're a long way from herd immunity. As I indicated, it'll be a complete disaster to try to achieve herd immunity through infections. So we have to play it safe until we get a vaccine. We can't just let our guard down now. We found these major race ethnicity differences in SARS-CoV-2 exposure, which suggests that that racial differences in disease are likely due primarily, I would argue, but at least in large part, to differences in exposures. What are our next steps? Well, you know, we have a lot of them. We want to understand these differences between asymptomatic and symptomatic infections. We want to identify antibody types associated with protection versus disease. I haven't showed you, but a large part of our research program is dedicated to measuring antibodies in people hospitalized with this virus. And we have some very interesting data there, which maybe Mike Betts and others will share in some of the talks later on this afternoon. We want to determine the longevity of this antibody response. I think this has honestly been overplayed a little bit. You know, it's typical in a viral infection like this to see a high antibody titer early in the response, and for it to sort of tinker down a little bit and then remain at some level, and that appears to be the case here. And I actually am not very afraid about this longevity issue. I think that obviously we have to be cautious as we communicate science, but to the general public. But I think this is a case where scientists might have been a little bit too cautious. And there's a lot of folks out there that are really doubting the protective ability of antibodies. We don't know. We need to do the studies. They're ongoing, but I fully expect that these antibodies, as I showed, they correlate very high with neutralizing capability in vitro. I fully expect that these antibodies are going to provide some sort of protection, and it looks like they'll provide protection for long periods of time. We are very interested in understanding how prior coronavirus infections shape immunity to SARS-CoV-2. This is actually a major part of my lab right now. We've been studying this in the context of flu, and we have some very interesting data as this relates to SARS-CoV-2. And yeah, monitoring seroprevalence. I think this is going to be a huge one moving forward as we have second and third waves in cities across our country and the world. We certainly need to understand the racial and ethnicity differences in seroprevalence. I think that this is crucial. In my mind, this is probably the most important thing that we have found and something that we have to address and put in place measures to reduce these disparities. Finally, we're developing serological assays. Hey, that vaccine is going to be around the corner, right? And so we need to develop serological assays that can distinguish infection versus vaccine-elicited immunity. And we're very interested in using these assays to look at seroprevalence rates and, again, demographics, which demographics are associated with high increases in vaccine-induced immunity versus infection-induced immunity. So I have a lot of people to thank. Ian Frank has set up that healthcare worker study with us. Deb has been very involved. John, Mike, Nuala, Dan, Joellen, everyone involved with either collecting samples and processing samples. Karen has been a co-PI on the pregnancy study and has just been fabulous to work with. Paul Bates and Sarah Cherry have done the in vitro studies. And here are all the folks from my lab. I tell you, I've been fortunate enough to be able to work at home during this pandemic. And my crew, who are one special technician here, and then the rest trainees who have come in every day to actually set up these assays. And not only have they been doing the technical side of things, but these are smart folks who actually figured out how to get this done. And I'll be forever thankful to them. So thank you for your attention. I think we might have time for one or two questions.
Okay. How many of the seropositive pregnant women were symptomatic?
Yeah, that's a great question. We are going through those data now to try to understand that. So anyone that is delivering at the hospitals now, they get mandatory NP swabs. And so not only are we looking at symptomatic versus asymptomatic, but we're also looking at correlations between NP positivity. And as you would suspect, many of the seropositive women were also NP positive. And we still need to analyze the data and see how that breaks down to symptomatic versus asymptomatic. That number is changing now. You can imagine in the middle of the pandemic and very early in the pandemic, seropositivity correlated very well with NP positivity. And that's because the virus was only around for a short amount of time. As we go through the study, someone that gets infected today and then gives birth, in September, it's likely they're going to be NP negative. But yeah, I agree that's something that we need to look at. In the healthcare worker study, it's about a 50-50 split between symptomatic and asymptomatic.
What is the false positive, false negative rate on the ELISA assay?
Yeah, so the false positive rate is less than 1%. So I think it's something like 0.5% for IgG. And false positive is sort of a misnomer, because what we think are that there's a small fraction of people that were walking around this planet before the pandemic that actually had antibodies that could cross-react. They're rare folks, but we think they are true antibody signals. So there's about 0.5% of us had those types of antibodies. We're very interested in that part of it, but from a purely technical standpoint, that gives about a 0.5% false positive rate. As far as false negatives, that's really hard to define. Out of our plasma transfusion cohort, I think it's less than 10% that had confirmed PCR, confirmed infections, but then went on to have no antibody titer in this receptor binding domain ELISA. We've then characterized these individuals further using ELISAs with full-length spike as well as N protein, and some of those just simply had antibodies against some of these other proteins. Florian Kramer's group at Mount Sinai showed that if you just wait a little bit longer, if you wait instead of 3 weeks after infection, another 2 or 3 weeks, that a lot of those seronegative individuals end up seroconverting and becoming positive. So some of that might just be a kinetic thing.
Quick question. In your opinion, would someone with a positive serology test need a vaccine or want a vaccine? Yeah,
that's a great question. I mean, nothing can beat natural immunity in most cases, right? So Yeah, I don't know. I think we should probably save those vaccines for the immunologically naive. That's what I would say. I think we all want to be antibody positive, right? The problem is if we become antibody positive through infection, that there will be just so much disease and death. And so, yeah, if we're one of the lucky ones that managed to get infected without having a lot of disease, we have to be cautious in communicating those results, as I indicated, but there's no reason to think that that wouldn't be protective.